you, Janine. And thank you all for attending this symposium. My name is Aki Nicolaitis. I'm super excited to get to be here today. I guess I'm just click. This guy? Okay, thank you. So yeah, today I'm going to talk to you about joint reliability and its relationship to reproducibility in human neuroscience. To start, oh, I see, okay, the other green. I am looking for collaborators, so if anyone's interested in the stuff I'm going to talk about today, hit me up. Let's chat. I think that this space has a lot of room, and there's a lot of opportunities for us to work together. So whenever talking about problems in the field or something like that, I think it's really always important to start with some of the successes. And I think it's really important to note that we've made huge strides already. And a lot of that is thanks to the people in this room and the OHBM community. And I think that is definitely worth celebrating. And we should definitely be giving ourselves a pat on the back for that. For example, we've moved to bigger sample sizes. We have these massive samples now. That's fantastic. We've moved from overfitting, univariate OLS that tends to overfit, to multivariate models with regularization. We are finally tackling this really complicated problem of how to do data aggregation in a way that is unbiased. And we've made significant strides in improving MRI reliability. From short scans to much longer scans, reaching up to reliability levels of like 0.9 or more for structural data and 0.8 for functional data if you have enough of it. And on the back of this successive series of wins, it kind of raises the question, like, are there other areas that we might be able to be improving even further? And to answer that question, I'm just going to jump into a bit of like psychometrics 101. So just to, I don't know how to point, but basically the left, oh, maybe this fancy thing. The left here, the observed correlation that you see in your study is a function of the population correlation between X and Y and the square root of the reliability of X and the reliability of Y. And this is joint reliability, and that's what I'm going to talk about today. A couple key points that come out of this, equation are that effect sizes are going to be maximized when the reliability of our variables are close to 1. And the further that we get from reliability equal to 1, the further away our observed correlations on average will be from the true underlying Bayesian behavior associations. So just a couple things for us to keep in mind as we go through this. Which brings up the question, would it be possible to improve the reliability of our behavioral measurements? Before jumping into that, it's important to note that there are some behavioral measurements out there that actually have pretty good reliability. Or rather, they have published studies that show that their reliability is quite good. Now, I'm going to just briefly go over a couple points that may bring up some issues of why we might not necessarily assume that these reliability or other psychometric estimates will be generalizable in all samples. And especially in the context of large-scale neuroimaging studies. But if we look in more detail at some of the, say, for example, diagnostic measurements that we have, there's a long history of studies that have shown that the DSM doesn't do great, and the SCID, the Structured Diagnostic Interview for the DSM, does a bit better, but not by much. This issue continues in the area of psychology as well. Obviously not a complete list, and summarizing the entirety of psychology would be impossible here. But each of these represent the mean of a series of many different assessments that were taken in each of these different studies. So you can see that the reliability of high-level reliability of 0.9 is still something that we can strive for in much of our behavioral measurements. But that's not all. There's other issues that are quite concerning, and I do not have time to get into them, but maybe this could be something we talk about in the Q&A or over drinks at some point if we want to. But for now, let's think about this issue. So if we have this question of maybe our behavioral measures are not as reliable as maybe we were expecting or hoping or theoretically could achieve, what is the impact of this reliability, lack of reliability, on our neuroimaging studies? Just to remind you, this joint reliability, right? It's like the ICC, for example, of your brain variable times your ICC of your behavioral variable. So let's take this, mock-up scenario. This is when we have we've idealized our imaging and we've achieved perfect reliability, and there's a true underlying correlation of 0.3 here. In this simulated data set, it's based on the normative model of the healthy brain network. What do we see? As we improve the reliability of our phenotypic data, we see this strengthening of effect sizes, right? We start to approach our true underlying phenotypic or a true underlying population level correlation. Now, this is obviously simulated data based on real data, but it's still simulated. So what if we actually looked at some real MRI data? Okay, well, in real MRI data where we have 2 structural scans and we have this true underlying relationship, which is really just the average across the 2 structural scans, we see similar sort of trend, right? Where we improve the reliability of the phenotypic variable and we see this approaching the true estimated effect size. Now, in this case, we're looking at IQ, right? So actually, the arrow could actually even go the opposite way. What we're actually doing here is we're not improving the reliability of the data. We're actually degrading the data to show the same basic point, just in reverse. So what could we do if we actually improved the reliability of our data? What would this look like? Well, in imaging, we have a pretty good handle on how to improve the reliability of our data. We've been talking about this for a long time. Get more data, right? So taking a look at the Philadelphia neurodevelopmental cohort, for example, and the colors don't come out quite as nice, but basically what you can see here is connectivity matrix of the Schaefer parcellation from 1,000 participants from the PNC, looking at correlations between the functional connectome and age, right? For 5 minutes of functional connectivity data. Okay, when we get 30 minutes of functional connectivity data, all of a sudden all of those associations with age start to come out a lot more coherently, right? And if we look at the distribution of those associations with age, we see that the zero, right, the relationships that are pretty close to zero go basically to null, they go down quite a bit, and a lot of the edges become a lot stronger associated with age, right? What if we could do the same but improve the reliability of behavioral measurements? This is kind of the key, the million-dollar question, right? How do we do that? There's a lot of different ways, it turns out, and a lot of people have talked about this. There's a lot of different ways that we could be improving the reliability of behavior measurements. Not going to go really into any of these, except for this one, deep phenotyping. Collect more behavioral data. What would that look like if we were to be able to do that? Now, it's actually pretty difficult to do that with most behaviors for a variety of reasons, but in already collected data. But the nice thing about actigraphy, if you have enough of it, is that you can kind of treat it like an fMRI time series in a similar way, where, for example, in the Healthy Brain Network, we have about 215 participants that have over 30 days of actigraphy. So that means that we have basically, they're wearing an actigraph, for those of you thinking about it, like basically an accelerometer and other things tracking your behavior. And a lot of people have investigated the relationship between motion and sleep and mood dysregulation and things like that. So it is definitely a clinically relevant behavioral phenotype. And if we collect more of this data, could we see improvements of behavioral reliability? Could we see concomitant improvements in our brain behavior associations? The answer is yes. So if we take a look here at this bottom triangle, we have 5 minutes of functional connectivity data with 4 days of actigraphy versus 30 minutes of functional connectivity with 15 days of actigraphy. We see both that the curve of brain behavior associations moves down and to the right, all becoming much, much stronger. This is again with the Schaefer atlas. If we look at every combination of functional connectivity and actigraphy data, we see this nice dose-dependent relationship with these 2 different types of actigraphy variables. The higher your brain and behavior data get in the reliability or the joint reliability of the 2, the stronger your effect sizes become. which begs the question, well, what does that mean for sample size? Right? Because if you are having weak samples or weak effects, you need a lot more samples to get to them, right? And in simulation data, that's pretty much what we're showing here, right? So if you can achieve high joint reliability, say in this simulation data where the true effect size would be a 0.2, you can get you'd be well powered to discover your true effect at a sample size maybe of 200. But if your joint reliability is lower, say like 0.2, you need thousands of subjects. Note that at some point, if your data is poor quality enough, having thousands of subjects is not going to help you at all. You know, a lot of us are focusing on precision medicine and thinking about, well, what's, you know, beyond the univariate case, right? This, for most of this, it's been basically like a psychometrics 101 applied to neuroimaging. The attenuation effect that I've discussed today extends to the case of machine learning as well. So if we were to do the same thing, say in a random forest, for example, we see that 15 days of actigraphy, that being the thing that you're trying to predict with your functional connectome, leads to very large decrease in the mean squared error compared to when you have only 2 days of data. It's obviously important when you're trying to do these kinds of assessments to make sure that they're working across different types of machine learning algorithms. These two are very different from one another, and we tend to see the same type of effect. Now, when we look at all 14 different types of actigraphy variables, we see the same general principle happen again and again and again. More data leads to much less error in our machine learning predictions, and that replicates both in the random forest case as well as in the sport vector machine case. So what have we talked about today, right? So we've seen clearly that low joint reliability is definitely presenting some kind of a serious challenge to us in human neuroscience. It's leading to both weak effect sizes and it's destabilizing the effect sizes and underpowering our studies, contrary to what we might be expecting our actual statistical power actually is. All of these in conjunction make it more likely that a single study could find a very strong effect that would be very exciting for a lot of us. But because of the destabilization of effect sizes, it increases the variance of the effect sizes in aggregate across the entirety of the literature. But given that it also is attenuating all the effect sizes, it means that all the other studies would fail to replicate because they would not be able to achieve that same level. I think implications for this are basically that we need to be thinking a lot more about data collection strategies that will allow us to get better behavioral reliability. For example, collecting more data per person, but that's not necessarily the only way. There's a lot that can be done on the individual task level to be optimized for different types of samples. Mobile data acquisition and deep phenotyping work that people are doing, I think, is absolutely essential and will be instrumental to solving this behavioral reliability issue. I think we also need to be putting more emphasis on the value of existing reliable data set, phenotypic measures, and I hope to have convinced you that prioritizing behavioral reliability will have a significant impact on improving the value of the neuroimaging data sets that we've collected. So thank you very much for your attention. I'd like to thank my collaborators at the Child Mind Institute and my funding sources as well. Please throw your questions or comments or any in the complaints in the app. Yeah, again, if these things interest you, please let's chat. I would love to see more people talking about behavioral reliability with me. So thanks.